Exhaustive search and progressive multiple sequence alignment approaches for doing multiple sequence alignment are good, but the problem is they are very computationally expensive techniques with respect to their time and memory conservations. So what is the solution? Again, solution is from sequence alignment techniques, where we studied BLAST and FASTA techniques, which were basically based on heuristics. So heuristic was our common day knowledge. And what was that information for BLAST or FASTA? That we say that instead of comparing individual characters in the sequences, let's try to compare blocks of those characters. So the third technique for multiple sequence alignment, which is block based MSA, it is based on blocks information. So instead of doing or comparing or aligning single characters, we try to compare blocks of these information. So what happens is that we have different blocks which are said to be segments in the given sequences, and then segments in the databases, and we try to find out alignment between these segments. So what we do, we have DNA sequences, protein blocks, and then RNA blocks. And what we do, we give query as a DNA, protein, or RNA block. Then we get information from the DB, which is also in the form of blocks, and then we retrieve our relevant results from that DB or database. So what is block based MSA? So first position of our given sequence is compared with the first position of first block. So if it matches, then it's okay. Otherwise, it tries to compare our first block with the second block and third block, and then so on. So wherever there is a match, it is retrieved. So advantage is that once we have got the block which is exactly matching, then we try to find out, or then we try to increase the size of that block and then see if it also works with the increased size. So what happens is, with this help, we can find out complete width of that block alignment, that how much width of the aligned block is there. So there are different tools for this block searcher alignment. Number one tool is block searcher tool. As the name implies, that it will try to find out different blocks between the multiple sequences. So how it works out? Again, it has a blocks DB where we have stored record of our protein or DNA sequences, and then we give a query as a block segment, and then we try to find out alignment between these two. So what will happen? It will try to find out all the related sequences from DB DB block against our given query. So the second one is our, again, what will happen? Effect of second and third blocks. What would be that effect? If first block matches, and then we will see, okay, second and third blocks are matching or no. अगर हमारे second और third block में match करते हैं, तो it will strengthen our belief that our first block matching or alignment algorithm was right. So second और third block का effect ये होगा कि हमें बेहतर alignment मिलेगी for our first block. Then what is blocks DB? Basically blocks DB जो है, हमारे पास एक प्रोजिट की डेटाबेस है जो प्रोटीन सीक्वेंसेस के लिए है. ब्लॉक्स डीबी उस प्रोजिट डेटाबेस की बेस पर जनरेट की गई है. सिमिलरली दिस इज कैलिब्रेटेड अगेंस्ट स्विस प्रोट डीबी, which is again a famous DB for protein sequences. So this PROSIT DB and Swiss prot, basically they are used for this generation of blocks database, which is used for block searching tool. So what is block maker tool? So basically block maker tool जो है हमारे पास, it has conserved blocks which are basically groups of 2 or more regions of protein sequences, and their inputs are basically in FASTA format, and then they have at least 2 sequences that should be present all the times in that blocks maker tool, that is compared against our given query, and the results are retrieved from our blocks database.